Thank you, Anna. Okay, so the lab, or what I'm going to talk about today, is really one of the approaches the lab is using, a systems level approach to understand and hopefully engineer plant stress responses. So I think many of you have probably seen this cartoon from me. I use it in almost every single one of my presentations. And I think it's just really a good introduction to something we all think about, which is how plants are responding or dealing with changes in their environments. It can be a stress, or it can just be any kind of signal or cue. It could be water, it could be drought, or it could be too much sun here. And so all of these responses typically occur through the central dogma that I'm showing here, DNA, RNA, protein. But of course, my lab is extremely focused on the RNA aspect of things and some of these RNAs that never translate into proteins. So we spend a lot of time looking at long encoding RNAs and a little bit of time looking at small RNAs and how all of this fits into the plant's response to this environmental cue. Too much sun, not enough water, how is the plant responding? And I think the ultimate question that many of us are trying to address is, can we improve upon this response? So today, or we kind of, in the lab, we deal with a whole number of, address these questions at a number of different scales, kind of at the genetic, single gene, trying to functionally characterize a gene. You heard from Liz yesterday, Liz's poster. She was looking at a specific long non-coding RNA. Matt had a poster yesterday looking at RNA modifications, these little lollipops that sit on RNAs and impact their translation and their structure. But today, Leon's going to be talking about, Leon here, a postdoc in the lab, is going to be talking about a systems level analysis to really integrate a whole bunch of data from the field to identify genes that are conferring stress resilience. And so we take this approach on a number of different plant species, cotton showing here, really because it's an important fiber crop, Waiuli because it's an important source of natural rubber, sorghum, again, sorghum's important for biomass, but also for grain, and then the mustards, really just because they're cool. Okay, and so, again, so Leon's going to be talking in the concurrent session in a little bit about some of this work. Okay, so what is Leon going to be talking about? And really, what is a systems level approach? What is an integrative approach? Well, Anna gave a really good talk in the earlier concurrent sessions about how you really integrate genomic information and transcriptomic information and phenomic information all into this like big kind of conglomeration of data, and you look for patterns. You try and figure out what is inducing something to ripen, or what is conferring resilience to a particular plant. And so here, for this experiment, we took 6 diverse sorghum accessions. So sorghum is kind of native to what we call the Old World, so Africa and Southern Asia. There are a lot of different land races that have evolved or been acclimatized to these different regions. And what we did is we just selected 6 of what were some of the most diverse accessions within this diversity panel that are not all necessarily really good at dealing with drought or with heat. So that's an important thing here, is that we're not looking for things that are all stellar performers under droughts, because we really need a contrast in this next stage of what we're going to be doing. So we take 6 diverse accessions. Then we expose these plants to stress in Arizona. Here's an indicative or a representative field in Arizona. I'm kidding. That's the white pockets in Arizona. A little grass trying to grow here. So we exposed these plants, these 6 accessions, just like what Anna was saying, in a kind of a standard randomized plot field design or block field design. And then we phenotyped, and I say we, this was a large project. We're along the ride for the genomics and the transcriptomics, but there was a large group of people working together to actually phenotype and sample how these plants, understand how these plants were growing. And you can see here just leaves taken at the end of the season, or close to the end of the season. There's a wide variety of responses to how these plants are coping with heat and drought stress in Arizona. And of course, these plants were grown under this what's called the gantry. It's a high throughput phenotyping facility at the University of Arizona, the Maricopa Agricultural Center. It's essentially the largest high throughput phenotyping, outdoor phenotyping platform in the world. And this minivan-sized phenotyping platform just runs up and down this field and allows for 3D reconstruction of the plants, as well as monitoring a whole bunch of other data points. So we intensely measure these plants. We collect a bunch of other data: growth rates, yield, photosynthesis, water use, metabolites, RNA. We already know the genetic differences, and I'm showing here. And then we try and integrate all of this complex data. So that's what Leon's going to be talking about, is how do we integrate those data? So that's a systems level integrative approach. And then, of course, we're hoping to use these findings to engineer resilient grains. So find ways that we can specifically target how these plants are transcription factors or other things that are regulating how these plants are responding to stressful environments. Okay, so I didn't get my updated slides in here. And that's okay, but I feel bad. But I feel bad because I was cribbing off of Jim, and he very nicely, and I should have thought about this, he very nicely thanked and had pictures of all of his lab people, and I was like, oh crap, I should have done that. You can just imagine, several of them are here in this room. They're very important people, and I'm very thankful for all of them. But please go and listen to Leon talk here in just a bit. Thank you.